Good morning, everyone. My name is Mishika Malik. I'm one of the neonatology fellows at Connecticut Children's, and my topic of research is impact of dietary carbohydrates on cytotoxin producing Klebsiella oxytoca, a potential pathway to necrotizing enterocolitis. Necrotizing enterocolitis is the most common and lethal emergency of premature infants. Bacterial colonization of neonatal intestine is a requirement for necrotizing enterocolitis. However, the precise role of microbes in the pathogenesis still remains unclear. Recent work, including the study performed by our group, demonstrated a predominance of proteobacteria in the gut of premature infants with NEC. Using 16S RNA sequencing, culture systems, biotyping, and PCR, our lab further identified Klebsiella oxytoca as a dominant microbe contributing to the dysbiosis in NEC pattern. Pathogenicity of Klebsiella oxytoca is dependent upon the production of potent cytotoxin called tillimycin, which in the presence of indole spontaneously converts to form a much less toxic product called tilivaline. The indole synthesis is done in a separate reaction as a byproduct of bacterial protein metabolism by the enzyme tryptophanase, also known as TNAA. This has been previously studied in E. coli that dietary sugars suppress the TNAA activity and indole production is reduced because many bacteria prefer carbohydrates over protein as the energy source. So the hypothesis of my study is that dietary carbohydrates diminish indole synthesis in Klebsiella oxytoca, thereby enhancing tillimycin production and increasing the propensity for intestinal damage. For methods, a toxin-producing KO-NEC isolate was cultured in lysogeny broth with and without glucose, and growth curves were generated by measuring the optical density at 600 nanometers. And you can see in figure 1, the growth patterns are pretty comparable when the toxin-producing KO was cultured in lysogeny medium with and without glucose. Then the supernatant indole and tillimycin concentrations were determined using rapid indole test and mass spectrometry. The supernatants were then applied to T84 enterocytes and subsequently stained with propylium iodide staining and analyzed by flow cytometry to quantify the induction of apoptosis. A toxin-producing KO-NEC isolate was also cultured in donor human milk, special care, and EleCare formula to see the effect of dietary sugars on toxin production. As you can see in figure 2, the indole production is inhibited when KO is grown in the presence of glucose. The figure 3 shows the flow cytometry histograms after incubating the T84 enterocytes in LB medium with and without glucose. The apoptotic cells have fragmented DNA that leaks out of them, causing them to be smaller in size, represented by sub-G1 population on the histogram. And in the presence of glucose, the apoptotic sub-G1 population was 53% as compared to 16% in the medium without glucose. Figure 4 shows the growth curves of toxin-producing KO in various dietary sugars like lactose, corn syrup solids, and human milk oligosaccharides seen in donor human milk. There's ongoing experiments in the lab currently looking at the effect of dietary sugars on toxin production of KO. So to conclude, carbohydrates decrease KO indole synthesis, enhance tillimycin production, and increase KO-induced enterocyte apoptosis. We speculate that in infants colonized with toxin-producing KO, an increase in the luminal carbohydrates, as would occur with carbohydrate malabsorption, could increase KO pathogenicity. Thank you.